All right, in this section, we're going to review genomics and gene therapy. So genomics is the study of genomes, or genes. The Human Genome Project was responsible for sequencing the human genome in 2003. The human genome contains 3.4 billion bases, and 99% of these bases are identical in all humans. So 99.9% of all human DNA is exactly identical from one individual to the next. The average functional gene contains 3,000 bases. There are 23,000 functional genes in humans. Genome size is not proportionate to the number of genes, and it does not really correlate to the complexity of the organism. Less than 2% of the human genome codes for functional proteins, so the remaining 98% may produce RNA molecules that help regulate the genes. So it's not necessarily about the number of genes that we have, but the amount of information that is contained in there. The RNA that helps efficiently separate out and divvy up that genetic information. So functional genomics is just simply how do our genes function. It's been responsible for identifying the roles of intergenic or those non-coding DNA sequences, those regions that those intron regions, or those regions that I was once taught were simply junk DNA. DNA sequences, so those intergenic DNA sequences are responsible for producing small RNAs that are involved in upregulating or downregulating particular genes. Comparative genomics is comparing the human genome to that of animals. It's used to determine how species evolved and how the genome functions. So things like the DNA sequence of chimpanzees and humans is 95 to 98% alike. So the human genome is, human genome is also 85% similar to that of a mouse. This is why mouse models are so frequently used when testing drugs and gene therapies. It's because their genome is so similar to ours. Proteomics is the study of structure, function, and the interaction of cellular proteins. The human proteome is just all of the proteins that result from the translation of all the coding regions of our genes. But as we've mentioned, one gene could code for up to 1,000 different proteins depending on how those introns and exons are spliced. Computer modeling of the 3D shape of proteins is an important part of proteoeconomics. If you have any interest at all in computer modeling or three-dimensional software, there is a huge pull for that in proteomics and genomics, in bioinformatics. Bioinformatics is just using a computer to study the genome. I knew many individuals in grad school whose entire thesis and work was based in bioinformatics. They never actually went to a wet lab. They never touched conventional tools. Everything they did was computer software, which is fascinating. It's a very interesting and rapidly growing field of science, is bioinformatics. This is just simply using computer analysis to find significant patterns in genomic information. Gene therapy is going to be the insertion of genetic material into cells for the treatment of a disorder. So there are certain gene therapies that have been used to successfully treat different types of inherited blindness. Both ex vivo and in vivo gene therapy methods are going to be used, and we're going to talk about each one of those. Ex vivo gene therapies are considered outside of the body. So in the case of an ex vivo gene therapy, the cells are going to be removed and then treated and then reinserted back into the body. So this has been a really effective method for treating the SCID disorder, so severe combined immunodeficiency disease, hemophilia A, Alzheimer's, Parkinson, Crohn's disease. A few cancers have all been treated this way. A lot of times leukemia and bone marrow disorders are treated in ex vivo, where the bone marrow is removed, irradiated, flushed with new genes, and then returned to the individual. In the case of in vivo gene therapy, therapeutic DNA is going to be injected straight into the body cells. A lot of times we're going to use a virus as the vector that carries the corrective gene, which is pretty fascinating. If you remember back when we talked about pathogens, we talked about viruses, and the way viruses work and replicate is by injecting their DNA into a host cell and then using that host cell to copy and replicate their own DNA. So viruses are phenomenal vectors for carrying gene therapies. They're already designed for injecting foreign DNA into host cells. So frequently viruses are going to be used as transporters. So if you remember back when we talked about cystic fibrosis patients lack a gene for a chloride transporter. They don't have the proper, they don't have the chloride ion channel that's going to allow chloride ions to pass through the cell membrane. With the chloride ions follows water and salt. Water and sodium are going to follow those chloride ions. This is going to keep the mucus found on your lung epithelial cells nice and light and fluid. This is going to allow your lung epithelial cells to beat the way that they should. It allows the cilia to beat. It allows them to circulate the mucus, and allows them to perform the way that they need to. So patients that are lacking that particular gene can be infected with a virus. The virus containing the functioning gene is either sprayed into the nose or injected directly into the respiratory tract. Now, the problem with in vivo gene therapy is that the effects don't really last as long as ex vivo gene therapy. You're not really producing you're not producing the DNA yourself. Temporarily, your cells the cells that have been infected with that particular gene will produce that gene. They'll produce that gene product. They'll produce that chloride ion channel. But when those cells die, they won't continue to pass on that information to the following generation. So the treatment is ongoing. So the effects don't really last as long as ex vivo therapy, but it has been used for the treatment of cardiovascular diseases, endocrine disorders, and Huntington disease. You should have a basic understanding of genomics, what the study is, what the purposes are, and know the basic differences between genomics and proteomics. Genomics is the study of genes. Proteomics is the study of our proteins. And then take a little bit of time. Go through gene therapies and why they might be used over traditional medications, but list some of the advantages and disadvantages to using gene therapies, to both those ex vivo and in vivo gene therapies. And let me know if you have any questions or comments concerning gene therapies. I'd love to discuss this further with you in our live discussion meeting.